Welcome to Rapid High Resolution Microfluidic Separations for Nucleic Acid Analysis. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Analysis of nucleic acid integrity to improve gene expression data or assess PCR fragments for resequencing applications has traditionally required costly and labor-intensive, difficult-to-scale, gel-based electrophoretic separations. This has hampered quantitative DNA and RNA analysis for a long time. The good news is that this is now changing thanks to microfluidics technology. A high-throughput alternative to electrophoretic separation for nucleic acid analysis in genomics research and next-generation sequencing is now available. The speakers in this webinar will introduce that new technology and describe how it has been successfully integrated into their workflows at two core genomic facilities. First up is Rick Bunch, Portfolio Director of Microfluidics at PerkinElmer. Rick will introduce PerkinElmer's LabChip GX automated electrophoresis system. Following Rick, Tumi Gatouche, Director of the Oncogenomics Core Facility at University of Miami, and S.B. Angiano, Manager of the Genomics Core at Baylor Institute for Immunology Research, will talk about how they've used this platform at their facilities. Rick, before I pass the microphone to you, I just want to encourage the audience to submit questions for all of us to answer at the end of the presentations. We'll answer as many questions as we can. Simply type your question into the Ask a Question box and hit Submit. Okay, Rick, let's get started.
Thank you very much, Tamlin. My name is Rick Bunch. I am the Portfolio Director for the Microfluidics business at PerkinElmer. What I'm going to do is try to frame the LabChip technology for the audience in about 10 minutes up in front of our application speakers. In this way, you would appreciate the kind of the core technology against which the frames of the applications from Tumi and SB are set. So we're going to talk a little bit about the GX product. The GX product is our LabChip entry, which analyzes nucleic acids. What we'll do is look a little bit at just kind of the foundation of microfluidics technology. How does this technology get embedded on a small 2-inch by 2-inch chip? We'll talk a little bit about the elements of the instrument, being the LabChip GX, and also follow up with a little explanation about the mechanics, the fluid mechanics of the chip and how it works and where it fits within the nucleic acid workflow. So microfluidics is a concept to miniaturize both the benchtop space in the laboratory, since the instruments themselves are very compact. The instruments themselves operate, again, on about a 2 by 2-inch microfluidics chip, which is where all the chemistry occurs. In addition, what's miniaturized is the chemistry that runs on the chip. Since we're dealing with microfluidic volumes, typically hundreds of nanoliters or picoliters, depending on the chip, the chemistry not only is small in volume, but can be accelerated to very high throughputs. The chips get manufactured by a process that was used and really perfected in the electronics industry called photolithography. On our chips, we use a glass substrate. The glass substrate is assembled in two pieces. One has the photolith etching, which are the channel patterns that you can see if you hold a chip up to the light just right, which connects together the buffer and reagent reservoir wells on the top of the chip on the component we call the caddy, which you'll see introduced in just a few slides. There's a top plate, not unlike a slide cover plate for those that have dealt before with just typically 1-inch microscope slides. The difference in this case is that there's a bond in between the two layers, such that the top layer and the etched layer are permanently bonded together. There are two different families of chips. The LabChip product from PerkinElmer uses what's called a zipper chip technology, which is represented in the left-hand side of this slide. There can be 1, 4, or 12 different zippers incorporated on the underside of a microfluidic chip. These are used to aspirate the sample directly from a microtiter plate well and introduced into the chemistry assay. You also may be familiar with other microfluidic technologies that are available on the market, such as the Agilent bioanalyzer, which uses a planar chip technology. Those chips are also produced by PerkinElmer. Agilent is one of our customers, commercial customers for the microfluidics technology. The microfluidics product line at PerkinElmer consists of two products for gel separations, the GX, which we'll be talking about today for nucleic acids, the GX2 for proteins, and also there is a product for called EasyReader for small molecule drug discovery separations. The LabChip XT is also a microfluidics product, but one of a little bit different nature. Its assays are preparative in nature. We won't be talking in detail about the XT as it applies to the nucleic acid space, but I'll just mention up front that it is a product which does fractionation and extracts selected fragment sizes from a nucleic acid sample and allows those to be collected at the end of that preparative assay for subsequent processing. We're going to concentrate today on the LabChip GX. So the LabChip product line is a combination not only of instrument but assay kits and software. There's a complete set of nucleic acid assays which are depicted in this next slide. The assay kits range in size of analysis from about 25 base pair fragments all the way up to 12,000 fragments. The kits may be selected for specialties such as pico levels of sensitivity in both RNA and DNA or size across the DNA fragment size spectrum. Recently introduced are assay protocols that enable not only the traditional users of LabChip to operate in a high throughput capacity, but also to give those same economies of scale to small batch users for these DNA assays. DNA assays with sample batch sizes as small as 20 to 24 wells can be economically processed. The DNA analysis on the LabChip GX system has far-ranging applications, two of which we're going to talk about today in subsequent speakers. But by example, those might be just simple PCR QC, might be genotyping, there might be some restriction digest analysis, or other nucleic acid analytical assays typically found in the laboratory. There's a sequence of a couple slides that I'll use to orient the viewers for how the chemistry moves across a chip. There's a combination of voltage separation, electrophoretic separation, and pressure applied to the chip. The blue on these chips depicts buffer wells, which are used to conduct the voltage from the instrument electrodes to the chemistry, and dye and marker, which is depicted here in red. The sample's aspirated up through the small cannula, which is attached to the underside of the chip, aspirated by pressure, depicted with the violet well in the back left-hand corner of the chip, is mixed then with the marker and the dye, pulled toward the vacuum well, and then reversed across an injection junction by a voltage applied across the buffer wells, which is depicted in the slide. Once the sample has been pulled across that injection junction, the voltage on the instrument is switched such that the sample, which is, in this case, nucleic acid, is pulled into a separation channel and separates like a traditional agarose gel. In this case, the separation continues and the dye is constructed such that it fluoresces under the presence of a laser. We get a laser-induced fluorescence, and then because of calibrants on the chip, can quantitate the size of the fragment and the concentration of each fragment size of DNA. Typically within the next-gen workflow, the GX is used for QCing the steps such as shear or size selection in PCR. Our subsequent presenters are going to go into more application detail about how the LabChip GX is used in their own application.
And yes, they are. But before we go on to those case studies, I just want to remind listeners that they can submit questions at any time by typing a question into the Ask a Question box. Tumi, we're ready for you to share your experiences with the LabChip GX.
Okay. Thank you, Tamlin, and thank you, Rick. My name is Tumi Getush. I'm the director of the Oncogenomics core facility of the Sylvester Cancer Center. I'm also the director of research and development of the Hassman Institute of Human Genomics Center for Genome Technology. Before I start going into the GX or in the LabChip GX, I would like to give you a short or brief overview of the institution that I work for. So the Hassman Institute of Human Genetics, the Center for Genome Technology, and the Sylvester Cancer Center, the Oncogenomics core facility, have a partnership between the core facilities since 2009. And between the two facilities, we carry out most genomics applications, genotyping, gene expression, capillary sequencing, and second-generation sequencing. Second-generation sequencing I call next-generation sequencing, what everybody calls next-generation sequencing or high-throughput sequencing. At our institution, we work at many different diseases. Here's an overview of the different diseases we work on, so multiple sclerosis, cancer, Parkinson, and Alzheimer's disease, to name a few. In terms of next-generation or second-generation sequencing applications that we run here, we do whole exome sequencing, targeted resequencing, whole genome sequencing, all the applications for RNA-seq and also ChIP-seq. So we run basically all the next-generation sequencing applications. And we are core facility, so we take samples from inside the institution as well as from outside the institution. During our work with the next-gen sequencing and also other genomics applications, we do a lot of quality control. So I've listed here a couple of, or basically almost all examples where we do quality control. So we do RNA quality control, measure the Win value from fresh as well as formalin-fixed paraffin embedded sources. We do RNA-seq QC. We library quant next-gen sequencing libraries, except, for example, the ribosero method. So we don't do it for every library, but for a lot of them. We QC microRNA, cRNA generation. We QC FFPs, DNA derived from FFPs. We QC also exomes, whole genome sequences, targeted captures, and ChIP-seqs, basically the libraries there. And we also QC PCR products and genomic DNA. This slide shows you a basic outline of the workflows of the Bioanalyzer 2100 and the LabChip GX. As you can see, for the most part, the workflows are pretty similar and straightforward. I don't have time to go into details here, but this is just to illustrate you that the procedures are very similar. In our facility, we QC a lot of different samples, often multiple 96-well plates. Here's a comparison of the process time for 96 samples using a Bioanalyzer and a LabChip GX. Please note that in the time comparison, I always assumed that the longer times for the LabChip GX, I always took the longer of the times for the LabChip GX, and the shortest time that I mentioned here for the Bioanalyzer. And you can see that you end up with substantial time savings running the GX versus the Bioanalyzer when you run 96 samples. Our most common NGS application is targeted resequencing and exome sequencing. We have automated this procedure on a Cyclone NGS. Here's a breakdown of the steps that we do on the Cyclone NGS. We QC 3 times using a Caliper GX. At the end of the shearing, after the amplification of the adapter ligated library, and at the final product before we load the product on the high-seq or the next-gen sequencer. This is again a workflow summary, just as a flow chart, and you can see that the 3 QC steps are done off the instrument. So here's how much time we save using a LabChip GX versus a Bioanalyzer when we do the next-gen sequencing library preparation for targeted or exome capture. We save 11 hours in processing time and 5 hours in hands-on time. I also like to mention that the cost per data point is significantly lower for the GX compared to the Bioanalyzer, about a third of the cost. So basically, you save a lot of time and also a lot of money using a LabChip GX versus a Bioanalyzer. Now I'm going to show you some data that was generated in our facility on the LabChip GX and the BioA using the same samples. For the quantitations I show you, I also added the results of a qubit measurement. Qubit uses fluorescent picogreen reagents to measure DNA concentration, and is generally believed to be the most accurate way of measuring DNA concentrations. The samples that I show are final targeted capture library products. So you can see the first slide here is a comparison of DNA concentrations. As I said, the bioanalyzer, which is in green, the qubit, which is in red, and the LabChip GX, which is in blue. And you can see that the values agree very well with each other, and there's very little difference between the three methods. Here's a comparison of the sizes of the library products measured by BioA and GX. The difference between the values determined by the two instruments is usually less than 10%, so they agree pretty well. You can see the sizes are about between 240 and 270 base pairs. Another example of NGS library products. Please note how well the qubit, the BioA, and the GX agree on the concentration measurements. I mean, they're almost right on top of each other. So these methods are extremely accurate in quantitating DNA concentration. And again, the same, comparing the DNA size measurements. Again, these are final products of libraries, and you can see how well they agree. I actually added the gel picture this time and also the electropherograms, and you see that the gel pictures and the electropherograms also agree very well in this slide, and also, I mean, in this measurement. And that's what we generally see. We see a very good agreement between the bioanalyzer and the LabChip GX when it comes to DNA library quant and also size measurements. And now I'm going to show you some other QC applications that we do in our facility. We use the GX for quality control, for FFPE samples, for both RNA and DNA, and you can see that the traces look nice. So this is FFPE DNA, and you can see that we can clearly see a larger size DNA measure. This is actually going to be much better once the genomic DNA assay is available on the LabChip GX. I think it's already there is an instrument out there that can do genomic DNA already, but if you can see this here, you can detect genomic DNA even with on the LabChip GX. We offer actually FFP DNA extraction and RNA extraction as a CLIA service. This is an RNA trace, also from FFP. You can clearly see that you see the products, longer product. You can resolve them on the LabChip GX. So you can run FFP samples on the LabChip GX, both RNA and DNA, no problem. Another sample of RNA from FFPs. And so to summarize my presentation, the LabChip GX positives are the throughput, which is very high, and it saves a lot of time using the LabChip GX when you have lots of samples. The cost per data point is lower than the bioanalyzer. And the analysis software is generally very well liked by our technicians and also by me. I like the analysis software of the LabChip GX very well. In terms of negatives, one negative that we have is the RNA size. On the RNA assay, the RNA size starts at 100 base pair versus 20 base pairs on the BioA, which is for some applications like the final step of Affymetrix microarrays, it might be a problem. And the minimum size of the kits is rather large, but as Rick explained before, they are coming out with smaller size kits, so that negative should go away. Generally, we start running the GX when we have 24 samples or more. And now to the acknowledgement slide, I'd like to thank our team here at the Hussman Institute for Human Genomics and at the Sylvester Cancer Center, the sequencing team, the capture team, and my oncogenomics core facility team. And also I'd like to acknowledge people from Caliper and Agilent that helped us with implementing these applications. And that's it. Thank you. This is a picture of the beach in Miami, so if you ever have time, come down here. Take care. Bye.
Thank you very much, Tumi. Espi, we're ready for you to tell us about your gene expression profiling workflow.
Thank you, Tamlin. My name is Esperanza Anguiano, and I'll be presenting on the application of the caliper and the LabChip GX for quality assurance in gene expression profiling workflows in our genomics core. Our institute is located in Dallas, Texas, and this is a picture of our core facility. The institute is under the direction of Dr. Young-Yoon Liu. Our research at the institute is focused and centered around immunobiology, with research interest in the fields of transplant immunology, inflammation, and autoimmune diseases. biotechnology, genomic medicine, and development of cancer vaccines. We are part of several multi-center studies funded by various government entities, and as such are positioned as a Center for Human Vaccines, Center for Immunology and Biodefense, Human Immunology Project Consortium, also as Autoimmune Center of Excellence. So then the Genomics Core at BIIR, or the Baylor Institute for Immunology Research, services all these research programs, and our goal is to generate transcriptional profiles from various sample sources and provide data mining support to both internal and external collaborators. Our services reach over 20 external partners in academia, government, and industry. As I mentioned earlier, our focus is immunobiology, and as such, we study the immune system. We believe that the blood or the circulatory system is the pipeline for the immune system. Blood travels to the entire body and in and out of organs, thereby sampling the different environments. This is why our focus has been in the profiling of whole blood. The profiling of the immune arsenal includes various platforms, including proteomics, cellular assays, and genomics. My work in the core lab focuses in genomics, specifically in the RNA profiling that leads to the generation of fingerprints that are specific to human disease. These fingerprints are quite specific for each condition, as we can see in these images. And I'm not going to go into the details of this, but this is the primary goal of our work. But you can find more information about this by referencing the publication cited at the bottom of this slide. This now is part of the steps followed in our workflow for gene expression profiling. I will focus only in the sample processing piece and share with you how the LabChip GX has been helping us assess not only the quality of samples received in our core, but also assess the efficiency of our processes. Together, this information helps us minimize technical variation or batch effect, which is an area of big concern in the analysis of whole genome gene expression data. At the bottom, we have a general workflow used in this profiling, which includes RNA extraction, amplification and labeling of cDNA products, and then followed by detection using whole genome microarray technology or RNA sequencing technology, ultimately leading to the unique biosignatures that we're after. The utility of the LabChip GX is best illustrated by focusing on the challenges encountered in the profiling of mRNA molecules. These challenges include RNA sample quality and purity, mRNA stability, RNA degrades quite easily, DNA contamination. This is important to know in the appropriate selection of downstream processes, especially in RNA sequencing. The removal of repeat masker elements, such as ribosomal RNA and globin in the case of whole blood experiments. And then furthermore, the efficiency of the enzymatic reactions in the generation of cDNA will also contribute to potential technical effects that might mask real biological changes. In our lab, we measure the quality of every RNA sample and any of its derived products. This is, for example, the electrophoretic profile at the bottom of the slide. Expect that when total RNA and globin-reduced RNA are analyzed in the LabChip GX. The second electrophoretic profile is what we normally get when assessing cRNA or cDNA samples. We look at both to ensure good quality expression data. With the caliper system, we have been able to analyze samples from controlled degradation experiments and identify an acceptable range of RQS values of RNA quality scores that ensure quality results when using the poly-A structure for initiation of cDNA synthesis. The profiles of these samples are illustrated on graphs with RQS values of 7, 8, and 9. Less than optimal results are obtained from samples in the other graphs. RQS values 6, 5, 4, and 3. These are profiles illustrating degraded RNA. We also use the caliper system to identify excess cold purification of DNA that may affect downstream analysis. In these electropherograms, the gel image shows sporadic bands across the range of sizes. We believe those to be DNA fragments. And in the electropherogram, one can also see an elevated baseline between the 18S and 28S peaks compared to the sample at the bottom of the right-hand bottom that has little or no DNA contamination. Another application for us is the identification of samples where globin depletion has not been very successful. These two graphs are examples of profiles exhibiting globin peaks compared to the smaller graph on the bottom of in the middle of the slide, where the removal of globin has been quite successful. I am not showing this, but in a smear analysis report that is produced with the software, the presence of these peaks is identified in the report itself and is used for downstream analysis and identifying samples that could be potentially outliers during data analysis. The benefits of globin reduction in our work, focusing on whole blood, is well illustrated in this chart. This is data published by a lab in UC San Diego in 2010. Essentially, the number of detected genes is greatly enhanced by the application of globin reduction. Therefore, measuring the efficiency of this reduction across all samples is of great importance in the generation of comparable data. Amplification and labeling prior to hybridization on microarrays is also assessed using the caliper system. Here I'm showing examples of the range of amplification efficiency that we have observed. This is important to us since a low amplification efficiency can result in lower numbers of genes detected and vice versa. And to conclude, the LabChip GX is an important tool in our lab for quality assurance purposes, and the data obtained from this system is useful in the selection of the most appropriate downstream analysis. For example, cDNA synthesis, even the sample profiles. The sensitivity, high-throughput format, faster runtimes, and cost efficiency have made the Caliper GX a great tool to have at our core facility in the assurance of good quality data. And as an acknowledgement, this is my team at the core facility at the Baylor Institute for Immunology Research in Dallas, Texas. I want to thank everyone for organizing this webinar and also everyone listening. Thank you very much.
SB, thank you. Before we begin the Q&A session, I just want to remind everyone one more time about submitting questions for our panelists. Type your question into the Ask a Question box and hit submit, and we'll answer as many as we can.
We have a lot of good questions already. Thank you very much, and we'll answer as many as we can. Toomey, the first questions are for you. What's the volume of the sample that has to be added to the 96-well sample plate?
Yeah, so thanks for the question. It's 2 microliter for the regular assay and 6 microliter for the high-sense assay.
Okay. Are there any additional steps needed after finishing a 96-well plate before running a second 96-well plate?
Yeah, there's one step. The gel dye solution needs to be refreshed after 96 samples or 3 hours of inactivity. It takes about 5 minutes to do that.
All right. To me, this time the question is multi-part. So I'm wondering how this system works for detection of individual plasmid DNA isoforms ranging a span of 2 to 12 kb. Can the system do any fragment analysis for higher molecular weight than 12 kb?
So for the 2 and 12, so it will work for 2 to 12 if you digest the plasmid with a restriction enzyme. At least you can linearize it for up to, I think, 10 kb, 10 to 12 kb it will work. Anything above that, you will see it accumulate on top of the gel, and you will get a picture similar to what I shown you on the FFP slide, where you see the black band right on top. So if you have a larger plasmid, you will get bunching up on top. I think there's a genomic assay in the planning that will be able to handle those kind of sizes that are larger than 12 kb. But generally, if it's below 10, it should be easy to do, and you just have to linearize your plasmid. Same basically like in a agarose jar.
All right, and can the lab chip be used to assess mitochondrial DNA?
It's the same problem basically, or the same case as in the plasmid DNA. If you digest it with multiple restriction enzymes to get fragments, yes, as long as you keep the size below about 10. If you want to run the whole mitochondrial genome just linearized, you will just see it basically bunch up on top. So you'll see that something there, but you won't get an accurate size assessment. Again, hopefully they will come out with a genomic DNA assay soon, and then that can be assessed.
Thanks, Toomey. SB, I have some questions for you. The first one is, how many samples need to be run at the same time for the LabChip GX to be more cost-effective than the bioanalyzer?
Well, it's a direct comparison of essentially on the Agilent bioanalyzer, to run one sample, you're utilizing an entire chip that you cannot recycle or reuse. And so essentially one sample you can run on the lab chip from caliper, and essentially that is what you need to run to get already a cost benefit, as the lab chip is reusable, and then you can scale down or up according to the number of samples that you're testing.
Okay, and then what is the minimum number of samples that can be run at one time with the LabChip GX?
As little as one.
Okay, and all the questions for you have to do with the number of samples. Here's another one. How many samples a day can be run on a setup?
On a setup on an 8-hour day, because we run 96-well plate samples because that works most efficiently based on our workflow. And so the run time on the caliper is 2 hours. Essentially, very little hands-on time, so very comfortably you could run up to 3 96-well plates on an 8-hour day if you wanted to. We can leave another one overnight, so essentially 4. So about 384-well plates. But you also have the capacity to run 384-well plates. That takes a little bit longer, but essentially you can double that in a 24-hour day.
Toomey, did you want to add to that, or?
No, she answered the question very well.
All right.
Yeah, I would like to add to the question about the kind of the low number of samples. The way, if I might, the way the instrument is set up, once the chip is loaded for the day, you can run as few as one at a time, as SB indicated. A different researcher could run a small batch of samples, 8, 10, 12, 20 samples, until the capacity of the chip is used up and it needs servicing. Or, again, as SB indicated, you could run as many as 384 in one pass.
Thanks, Rick. Toomey, I do have a question for you. Another question. What is the advantage of this automatic system over other ones provided by competitors? Running costs for samples specifically?
Yeah, so that's the main advantage. It's the throughput and the cost per sample. So the throughput is significantly higher than any of the other competitor system, and the cost per sample is also significantly lower, about a third to a quarter of the cost per sample or per data point. So yeah, it's cost and throughput mainly what are the advantages. Also the software, compared to the older systems, the software is a little bit more user-friendly, but mainly throughput and cost.
Okay, Rick, I have some questions for you now. Can the system be used to assess small RNA composition in total RNA?
Yeah, so for small RNA, there's a limit to what you can see and what you can quantitate. Some very small RNA will actually kind of co-migrate with the lower marker. That's with respect to our current kit. We are looking at ways to move that lower marker so that researchers can use the RNA kits for small RNA.
Okay, and then what is the smallest size difference that can be resolved into a unique peak band? For instance, if I wanted to identify specific transcript variants after PCR rather than using a slab gel.
Sure. Everybody's interested in higher and higher resolution. The way we define resolution as our spec is being able to resolve two peaks that are separated by a low point in between the two peaks by 50% return to baseline. Based on that specification, our highest resolution kit can resolve on the order of 3 or 4 base pair resolution.
All right, thanks, Rick. Another question for you. What is the detection limit on DNA concentration, and what is the CV of the measurement on LabChip GX?
Yep, in general, the CV measurements for DNA are going to run about plus or minus 10%. There are 2 kits against which I can answer the question regarding sensitivity. Our standard kit, which is labeled a DNA 5K kit because that is its upper range, has a measurement sensitivity of about 5 nanograms per microliter. We do also offer a high sensitivity DNA kit, which extends that lower range down to about 10 picograms per microliter.
Okay, and now I'm going back to Toomey. Toomey, we have a lot of questions directed at you. Does Caliper, and this is a multi-part one, so hang on. Does Caliper LabChip GX, can it differentiate between DNA and RNA in one sample, and how could you make sure there's no crossover contamination between sample that is analyzed?
Yeah, so the assays, I believe, are using 2 different dyes. One uses a dye that binds to double-stranded DNA, and the other dye is binding to single-stranded RNA. Now, when you do RNA traces, you sometimes see high molecular weight bands that are typically DNA. You can, so they are way past the 28S band of the RNA, and there's unlikely any significant amount of material there when you run RNA. So you can sometimes see genomic DNA in there. SB, in her presentation, I believe she showed some genomic DNA that's between the 18S and the 28S peaks, and you can see those. I believe there you can see, if the DNA is degraded, you can see some DNA there. So basically, you can see both DNA and RNA at the same time. Sometimes it's typically for us, we don't want to see DNA. So if there's too much genomic DNA, we do DNase digestion and run the sample again. In terms of quantitation, yes, it throws off the quantitation a little bit, but not. It's hard to say how much it is. Depends on the genomic DNA contamination, obviously. In the RNA chip, in the DNA chip, I don't think we see a lot of single-stranded material. Maybe sometimes some small degraded samples at the bottom, like right where the detection limit is, like maybe 100 base or something. You see a small peak that might be something there, but typically you see more RNA, so you see more DNA in the RNA traces than the other way around. So for us, it's actually good to see that because we use that for QC measures. So as I said, if we see too much genomic DNA, we do a DNase digest. But if you really want to exclude it, then you're going to have to do a DNase digest anyway. Hope that answers the question. Okay.
I have one more. I think this is a simple question for you. If you can comment on this, what is the approximate cost per sample?
Yeah, I don't want to necessarily disclose the price that we pay because everybody pays different. But I can tell you it's about a third to a quarter of the price that we pay for like an Agilent bioanalyzer, for example. So when we do the math and we compare the prices, it's about a third to a quarter per sample of the bioanalyzer.
Thank you, Toomey. Thank you, SB, and thank you, Rick, for answering all those questions, and thank you, everyone who's listening, for sending in so many good questions.
I forgot to say one thing. I'm sorry. The third or a quarter if you run it at full capacity. So if you run 24 samples, obviously it's not going to be a third or a quarter. It's going to be about the same as a bioanalyzer. But if you run 96 samples or more, then it's going to be a third to a quarter. So sorry. I just want to clarify that.
No, I think that's good. Thank you, Toomey.
Okay.
And thanks again, everyone, for submitting such great questions. Our time's up. Thank you for listening in. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again or you can recommend it to friends and colleagues. I want to thank our panelists as well as our sponsor, PerkinElmer. We'll be sending you a survey shortly and hope you can take the time to respond. Thank you very much.
All right. Thank you.
Thank you.